we're going to continue our discussion of chemistry by looking at the large molecules that perform all the important functions in your cells. And these molecules are known as macromolecules. So we're going to delve into a little bit of organic chemistry. Imagine we could build a cell from scratch, right from elements, and throw them together and make a cell. If we were going to do that, we need something to build the backbone of the macromolecules that form the structure of the cell and that form enzymes, etc. So what element should we use if we were going to do that? Well, we have to have an element that is capable of forming strong and lasting bonds, because there are some molecules in your cells that stick around for a long time and they need to add structural support, for instance. But we also have some molecules that need to be taken apart and reformed frequently. So ATP is used to shuttle energy around in the cell, and it can be taken apart into ADP and a phosphate, an inorganic phosphate, but then stuck back together right away. And it might be taken apart and put back together many, many times in a second. Also, if you look at DNA, DNA has to be unzipped. It consists of two strands, and they have to be pulled apart in order for your cell to read the information that's contained within the DNA, and then the DNA molecule has to be zipped back up. So we need some sort of atom that can form long-lasting covalent bonds, but also form weak interactions as well. We need an atom that can form many bonds, so it can stick itself to many other things, and that allows for the construction of complex molecules. We need to build some structures that are impervious to H2O, so they don't dissolve in water. But then we need to make some others that are soluble in water, because most of the chemistry of life happens in water. So we need molecular backbones that are versatile, interchangeable, reusable. They can be taken apart for energy production. They can be used for storing energy. They can be used for replication, etc., etc., etc. Carbon meets all of these requirements. And the reason for that is because it has 4 valence electrons. That makes carbon very versatile. So it only has 4 electrons in the outer shell. In order to be stable, it needs 8. And that means that carbon is capable of sharing 4 pairs of electrons with other atoms. If it's sharing 1 pair of electrons with another atom, that would be a single bond. If it's sharing 2 pairs, that would be a double bond. 3 pairs, that would be a triple bond, etc. But we can get some very, very complex molecules, any kind of configuration or shape you can imagine, using carbon. So all life that we know of is built around molecules that contain a carbon backbone. It's been hypothesized that silicone might fit this requirement as well, because silicone also has a valence of 4, and perhaps out there in the universe somewhere there is life that's quite different from what we know, and it's based around silicone instead of carbon, but we haven't encountered anything like that yet. So the study of molecules that have a carbon backbone is known as organic chemistry. Organic chemistry looks at molecules that are found in cells typically, although there are some that are associated with non-living processes also. And there's a whole naming system for organic molecules based on how many carbons they have, and also based on what other things are attached to that carbon backbone. So at the top here we've got methane, and this would be the simplest organic molecule. We've got 1 carbon and it's sharing 4 pairs of electrons with 4 hydrogen atoms. So it's got 4 single bonds, 1 to each of these 4 hydrogen atoms. And you can see the structural formula there. That's kind of the shorthand for this. That's the way you would draw this out. Again, a straight line, just 1 line means 1 shared pair of electrons. We could use a ball and stick model. So if you've played around with chemistry sets in the lab, this is what you're doing. And then there's space filling models, which are a little more realistic, because remember the orbits and so on are going to overlap. The next more complex organic molecule on this list here is ethane. And notice that we also have the formula, of course, that tells us how many atoms we have in the molecule. But notice that the backbone here is 2 carbons, and then we just have hydrogens attached to that. When we just have carbons and hydrogens like this, we call that a hydrocarbon, which makes sense. Now down the bottom there, we've got ethene or ethylene, and notice it's a bit different the way it's structured. So it has, again, just carbon and hydrogen. There's only 2 carbons, but this time we've got a double bond between the 2 carbons, so they're sharing 2 pairs of electrons. And that means that we have fewer hydrogens attached. We have 4 hydrogens instead of 6. I won't get into the naming system in great detail, but I do recommend that at some point you try to learn it and understand it, because it will tell you a whole lot about the molecule. So for instance, meth, that first part of the name tells us that there's only 1 carbon involved. Eth, that tells us that there are 2 carbons. Pro, that tells us that there's 3 carbons, etc. The ane at the end tells us that there are no double bonds. There's only single bonds within the carbon backbone. The ene tells us there's a double bond within the carbon backbone. So these names all make sense, and they tell you quite a lot about the molecule. Organic molecules don't consist only of carbon, of course. There's other things hanging off of that carbon backbone, and sometimes there's carbon bits that hang off of the main backbone. But there's always going to be some hydrogen, and there's quite often oxygen and nitrogen, and sometimes other things like sulfur and phosphorus, but these are the big 4 here. So CHON, you should know the properties of those atoms. And if you know the properties of the atoms, and really all you have to know to do this is know the number of valence electrons. If you know the number of valence electrons, you can figure out what might happen when these atoms come together. Now what you're seeing down the bottom here is what's called a Lewis dot diagram. And we're just representing the electrons in the valence shell as dots. And what you can do is you can bring together atoms and try to fill up the shells of all the atoms. It's kind of a fun little puzzle. So here I've got some examples of some Lewis dot diagrams. So the big one at the top there is showing what happens when carbon gets together with oxygen, or one of the things that can happen. So we've got the oxygen electrons being shown in red, and we've got the carbon electrons being shown in black. So what we've done here is we've rearranged the dots so that everyone is happy and everyone has a full shell. So the row of electrons here represents a double bond because we've got 2 pairs of shared electrons. So altogether, we're sharing 4 pairs of electrons. And this completes the shell for oxygen because now it has 8 in its outer shell. Carbon has 8 in its outer shell as well. And just remember that this would be represented like this. Each one of these lines represents a shared pair of electrons. So that's carbon dioxide. Now down the bottom on the left, we've got water. And in this case, we're representing the oxygen's electrons as circles and the hydrogen's electrons as crosses or X's. This is an easier way to do it if you're doing this on a sheet of paper and you don't have 2 different colors. In the middle, we've got methane. So in this case, carbon has 4 valence electrons, hydrogen only has 1, but the way they're sharing things, everybody is happy, everyone has a complete valence shell. Remember, the innermost shell can only hold 2 electrons. So for hydrogen, it just wants to get 2 electrons in that shell. And then finally, we've got ammonia. Nitrogen has 5 electrons in its outer shell. It's looking to add 3. And it's getting those 3 by sharing electrons with hydrogen. So we have our backbone of carbon, just a long chain of carbon molecules, but of course other things attached to those carbons. In the simplest case, we just have hydrogen attached to a chain of carbons, and that's a hydrocarbon. But we can have various other things attached as well. And those bits that hang off of the main chain are known as functional groups or side chains. So I'm going to talk about these, and there's quite a few of them, even more than I've put into these tables, but I want you to know the ones that I have marked with the big red arrows. All of them are important, really, but the ones that I've marked are critical when it comes to understanding the 4 classes of macromolecules that we're going to talk about. It gives those macromolecules their special properties. First of all, we have hydroxyl groups, and a hydroxyl group is just simply an OH, an oxygen and a hydrogen. If we have this attached to an organic molecule, it makes that molecule an alcohol. So the example there, ethanol, eth means 2, so there's 2 carbons. All on the end tells you you're dealing with an alcohol. So methanol, for instance, would be quite similar. It would just be 1 carbon, but again with that hydroxyl. Now I should mention that many molecules have several different types of functional groups. So when it comes to naming them, they name them based on the one that gives the molecule its most prominent characteristics. So if we have a hydroxyl on a molecule, it might not necessarily be called an alcohol if there's other functional groups that are more important. Okay, the next group there would be aldehydes. I'm not going to have you remember that structure, but we've got this weird ending to the chain where the last carbon has this oxygen double bound to it and then also a hydrogen singly bound. The next one would be ketones. You've probably heard of ketones before. You've probably heard of the ketogenic diet, or keto as it's usually called. In ketogenesis, if your body is in ketogenesis, what you're doing is you're depriving your body of carbohydrates, and instead of using glucose as the main way to transport energy through the bloodstream to all the cells of your body manufactures these molecules that contain this keto group. And those ketones can be used as fuel instead of glucose. It's a rather effective diet, I have to say. I gave it a shot and lost about 30 pounds doing it. And there are probably some pretty decent health benefits from it, but it's not something we're going to talk about this term. We'll talk about that in more detail when we talk about nutrition in Bio 112. The last one on the page there is the carboxyl group. And this generates a class of acids. An example that you've probably heard of is acetic acid. That is simply vinegar. On to a couple more. So I should point out, in case it wasn't obvious in the last slide, that in the example, the part that's highlighted in white is the actual functional group. In the structural formula, the R represents the carbon backbone. So just assume in these 3 examples that the carbon backbone is going off towards the left, and it might be 1 carbon, it might be 20 carbons, but that's what that represents. Okay, so the first group, we have the amino functional group, and molecules that contain this are known as amines. And an example you might have heard of before is methylamine. So it's meth because there's 1 carbon, and then of course amine because it has this amino group at the end. If you've watched Breaking Bad, you might realize that methylamine can be used to make crystal meth. But you probably also recognize amino from amino acids. And that's why this one's important for us to know. All amino acids have that group. Next one would be phosphates. So you can see the white highlighted bit in the example. We've got this phosphorus with 4 oxygens attached to it. Very important functional group. It's found in nucleic acids, DNA, and RNA. And as we've talked about already, if we pull that off ATP to make ADP, we liberate a bit of energy as well. So when that's pulled off of ATP, the molecule is rearranged slightly, and the new bonds that are produced give off energy. At the end here, we've got sulfhydryl functional group. These are important when it comes to proteins. So we'll talk about the structure of protein in a bit of detail. Proteins can be very complicated. They can be twisted and folded and so on. And we can get these sulfur bridges, these disulfide bonds. We'll talk about that later, but they help support the molecule They help it maintain its structure. The example you're seeing there, mercaptoethanol, this is something that when it interacts with proteins, it actually breaks those sulfur bonds and helps break down and unravel the protein. It's something that's found in some really powerful cleaning agents. It has kind of a fishy smell to it. So now that we've talked about some basic organic chemistry, let's go on and look at the actual macromolecules. And there's 4 varieties. We have lipids, or fats. We have carbohydrates, which are sugars and also structural molecules as well. We have proteins, and we have nucleic acids, which are RNA and DNA. A few terms that we should go through first. A polymer is a large molecule that consists of several building blocks, essentially. You can think of the building blocks almost like Lego blocks. Let's say you stick 100 Lego blocks together. You've got, I guess, a polymer of Lego. So polymers are made up of small building blocks, smaller organic molecules that are all very similar. The monomer is the actual building block. So for instance, proteins are long chains of amino acids. The amino acid would be the monomer. The protein itself is the polymer. And all polymers are made the same way in your cells. It doesn't matter if we're talking about proteins, nucleic acids. In all cases, the monomers come together and a water molecule is drawn off as a new bond is formed. So here we have an organic molecule of some sort, a macromolecule, and it consists of, to start, 3 monomers, 1, 2, 3. And we're going to add another monomer to lengthen this molecule. Note that at the end of the polymer, we have a free hydrogen. At the end of the monomer that's coming in, we have a free hydroxyl. They come together and a water molecule is pulled off and a bond is formed between the polymer and the new monomer. This is called dehydration synthesis. Synthesis because we're making a macromolecule one piece at a time. Dehydration because we're pulling off water. If we want to break down a macromolecule, it's the exact opposite. We introduce a water and we break that bond that holds the monomers together. This is called hydrolysis. Hydro refers to water, lysis means to break. So the terms do actually make perfect sense. So now that we've gone over this, let's build a cell. So let's just imagine we're building a cell. We have these 4 different macromolecules. What role do those different macromolecules play? Remember that you are mostly water and your cells are mostly water, and all of the chemistry of life takes place in water. Water is essential. So we have cells that are made up of mostly water and they're surrounded by mostly water. So you have interstitial fluid between your cells. And your cell has to separate itself from its surroundings. Water is interesting, unusual, in that it's a polar molecule. Remember, that's because the electrons are shared unequally between oxygen and hydrogen, and that gives the molecule a dipole. It gives it a slight charge distribution that's uneven. And that means that water will grab onto anything that has a charge and it will solubilize it. So let's say we have a molecule that's got a slight positive charge to it. It's going to be attracted to the slightly negative oxygen of a water molecule, and then the hydrogens of that water molecule are going to interact with the oxygens of other water molecules, and basically that molecule is going to be dissolved or suspended in the water. So in order for the cell to not simply disintegrate in water, we need to have compounds that are not charged, that make up the membrane and make up basically the walls that are going to make up the compartments within the cell. And that's done by lipids. So lipids are formed by dehydration synthesis between glycerol and fatty acid. Lipids are kind of unusual compared to the other 3 macromolecules in that glycerol and fatty acid are quite different. If we look at proteins, like I mentioned, they're made out of amino acids that all have the same essential properties. But glycerol and fatty acid, those two components are a bit different. So we have glycerol, which is actually an alcohol. And if you look, we've got a chain of 3 carbons and we've got hydroxyls hanging off the end. Well, those hydroxyls can be used in dehydration synthesis to create a bond between the glycerol and a fatty acid. The fatty acid is a hydrocarbon. So you can see it's just a chain of carbons and hydrogens. Now remember that carbon and hydrogen have very, very similar electronegativities. And that means that the electrons are shared equally between the carbons in the chain and between the carbons and the hydrogens. So there isn't an unequal charge distribution, and that long fatty acid and the glycerol, after this dehydration synthesis has occurred, are hydrophobic. They don't interact with water because they don't have a charge. So this is a very stable molecule. And the one you're seeing here wouldn't be used to build a membrane. It would actually be used to store energy for long periods of time. So lipids consist of 2 parts. We have our glycerol, and then we have fatty acids, these long straight chains attached to that glycerol. Normally there's 2 or 3, but we can have variations of that. So we've got fatty acids that consist of a long carbon skeleton, usually 16 to 18, but that's quite variable as well. One end is a carboxyl group. We've got entirely non-polar carbon to hydrogen bonds and carbon to carbon bonds, of course, and that makes this fatty acid hydrophobic. The glycerol is also hydrophobic once it's entered into this arrangement with fatty acids. There are 2 broad categories of fatty acids. We can have some that are said to be saturated, and that's what you're seeing on the left, and we can have unsaturated. So saturated refers to the fact that that hydrocarbon contains as many hydrogens as it possibly can. So notice that the carbons kind of wiggle back and forth a little bit, but the chain overall is straight. It's just carbon and hydrogen. Now imagine that we introduce a double bond into the backbone of the fatty acid. In order to introduce a double bond, we have to lose 2 hydrogens. And you can see that the example here has 2 double bonds within the carbon backbone. And in each occurrence where we have a double bond, we're missing 2 hydrogens. What this does is it causes the chain to kink. It kind of upsets the electrons within the shell that are causing the double bond displace other electrons in that shell. We won't get into that in great detail, but just realize that it causes the chain to kink. So unsaturated fatty acids have kinks in them. We'll get back to saturated versus unsaturated in a bit, but first let's talk about triglycerides. Triglycerides are used to store energy long-term in your cells. There's other ways to store energy as well, but this is one of the most prevalent. So tri means 3. A triglyceride is a lipid that consists of a glycerol attached to 3 fatty acids. The fatty acids store a great deal of potential energy, and that's because all of the bonds within the fatty acid are non-covalent. And remember from our discussion of chemistry earlier, non-covalent bonds have a low bond energy. So those bonds between the hydrogen and the carbon and the carbon and the carbon are fairly easy to break. It doesn't take much energy. But when we combine the carbons and the oxygens that we pulled off with oxygen to make water and carbon dioxide, we get a lot more energy back. The picture that you're seeing there is showing fat droplets inside an adipose cell. An adipose cell is a fat cell. They actually start out very small, and as you store fat, they swell and swell and swell. So if you gain weight, you're actually not gaining cells, the cells are just expanding in size. Another really important group of lipids are the phospholipids, and as the name suggests, they have a phosphate group attached to them. We have 2 fatty acids, and they may be saturated or unsaturated, and they're attached to a glycerol. But then attached to that, we have something called a phosphate, and we have something else called a choline attached to that. The phosphate has a full negative charge, so it's got one extra unbalanced electron. And the choline has a full positive charge, so it's missing one whole electron. We have this region that consists of the glycerol, phosphate, and choline, and that's known as the head of the molecule, and it's hydrophilic, which means that it is attracted to water. And again, that's because water has a dipole and it will interact with those charges. The oxygen on a water molecule will bind to the choline, the hydrogens on a water molecule will bind to the phosphate. But the fatty acid region is hydrophobic because it doesn't have an uneven charge distribution anywhere. And down the bottom there towards the right, that's the way that this structure is usually represented and how you might draw it if you were to draw the membrane, for instance. That's where we see these lipids. So recall that a molecule that's hydrophilic is one that will interact strongly with water because it has a dipole or a full charge. A hydrophobic molecule is one that won't interact with water because it doesn't have an uneven charge distribution. An amphipathic molecule is a molecule that has both properties. So phospholipids are amphipathic. Amphi means both. Think of an amphibian. It can live in both the water and on the land. So phospholipids, the hydrophilic head will interact with water, the hydrophobic tail will not. So if you throw a bunch of phospholipids into water, they will rearrange themselves passively so that the hydrophilic heads are pointing towards the water and the hydrophobic tails are pointing away. It doesn't take any energy for these more stable configurations to form. So we can have what's called a micelle. That's where we get droplets forming, tiny little droplets. Or we can get a phospholipid bilayer. And as I mentioned, you can make your own phospholipid bilayer. You can make an artificial membrane by taking phospholipids, putting them onto the surface of water, floating them on top of water in a water bath, for instance, and vibrating it, shaking it. And some of the phospholipids will arrange themselves into this artificial membrane. Now, if it's an artificial membrane that you're going to use for research reasons or something like that, there's some other things you have to do to make it more resemble a membrane, but we'll get back to that in a bit. Okay, so as I've already alluded to, membranes are made up of phospholipids, and they form a barrier between the cell and the outside world, and they're also used to make compartments within the cell. So we have two layers of phospholipid. The inner core is made up of the hydrophobic tails, and the hydrophilic heads interact with water. And this is a very stable configuration. It doesn't require any energy to maintain that configuration. So because we've got a full positive and full negative charge on the heads, they are going to be attracted very strongly to water. And the tails aren't really being pushed away by the water, they're just being excluded by the water, and they're forming interactions with themselves. The tails, the fatty acid tails, because they're quite long, there are actually van der Waals forces between adjacent tails on adjacent molecules that help hold that structure together. So this gives the cell protection from the external environment, and water, like I said, is actually potentially a destructive force. It will solubilize things, it will dissolve things. And also it allows the cell to make compartments inside, so things like the Golgi body, the endoplasmic reticulum, the nucleus, etc. On to our next macromolecule, and that would be sugar, carbohydrates. Sugar is also referred to as saccharin. It's a term that's not used very often in English, but let's say someone reads you a really sappy, sweet poem, you might say that poem was very saccharin. Anyway, a saccharide is a sugar. We can have polysaccharides, which consist of many sugars stuck together. But the actual sugar itself, the monomer, is generally 3 to 7 carbons in size, and usually it's a ring-shaped structure, as we'll see. It has a carbonyl group at one end, and it has an alcohol group as well. So here are some typical monosaccharides. So these are the monomers that make up larger carbohydrates. So we have some triose sugars that consist of only 3 carbons. We have pentose sugars, which are more common. Ribose you've probably heard of. That is in RNA, ribonucleic acid. Deoxyribose is very similar, and that's found in DNA. And then we have the hexose sugars, which are things like glucose and galactose and some of the other sugars you're more familiar with. Notice down the bottom we have ketone versions of that. So we have fructose, which you've no doubt heard of, high fructose corn syrup, for instance. So corn extracts contain a lot of fructose. And it's similar to other hexose sugars, except it has this ketone group in there as well. So monosaccharides can exist in a straight or linear form, but generally they're bent into a ring. Notice on the first diagram there on the left, we've got the linear form, and notice that the carbons are numbered 1, 2, 3, 4, 5, 6, starting with the carboxyl. We won't talk about that in too much detail, but if you're wondering when you see these diagrams what the numbers mean, that's what they mean. So generally, monosaccharides are bent into a ring, and we call that a cyclic form. So we get bonds forming between the ends of the molecules, and in this case you can see that carbon number 6 is actually not part of that ring, it's outside of the ring. Down the bottom there, part B, you're seeing the abbreviated ring structure, and this is how biochemists and organic chemists would draw this structure. If you see an intersection there and it's got a number, you can see it doesn't actually have a C, it's just understood that at those points there's a carbon. Monosaccharides can come together through dehydration synthesis, just like other macromolecules, and we get a glycosidic linkage forming between the monomers. The numbers there, 1 to 4, 1 to 2, refer to the numbers of the carbons that are involved in that linkage. So here we've got the creation of a couple of disaccharides. Di means 2. And a lot of the carbohydrates that we consume as food are disaccharides. So sucrose down the bottom, that's table sugar. And it consists of a glucose monosaccharide and a fructose monosaccharide stuck together. Maltose consists of glucose and glucose stuck together. And in fact, if you break down starch, what happens in the intestines is it's broken down into maltose, and then maltose can be broken down further into glucose. Carbohydrates can be used to store energy in both plants and animals. In plants, starch is one of the main ways that a plant can store energy long term. So for instance, a potato is a highly modified root or tuber, and it contains mainly starch. And that starch is made up entirely of glucose. So we can have amylose or amylopectin. Those are 2 different forms of starch. Note that amylose is just kind of a spiral of glucose molecules. Amylopectin has branches in it. But each one of those green hexagons represents a glucose molecule. In animals, we store glucose as glycogen. And we do that mainly in the liver and in muscle cells. And glycogen is a highly branched molecule, but again, it consists entirely of glucose. And if your blood glucose levels drop, then that stimulates your liver to pull off those glucose molecules and dump them into the blood. And your muscles can store glucose for times when you need that extra energy. In many organisms, carbohydrates are also used for structure. Plants have a cell wall that's mostly cellulose, and that cellulose is a carbohydrate. It's made out of glucose. The glucose molecules are stuck together in such a way that we can't break them apart. We don't possess the enzyme needed to break the glycosidic linkages that are found in cellulose, even though we can break glycosidic linkages in many other molecules. So what you're seeing down the bottom here, B, we've got starch, and it's got this 1 to 4 linkage. And then in cellulose, on the right, we've got this 1 to 4 linkage as well. But notice that the glucose monomers are oriented differently. So you can see in the first diagram, B, the hydroxyl group that's highlighted there, they're all down the bottom. But in cellulose, they alternate. And that gives this glycosidic linkage a slightly different shape than we see in starch. And it means that the enzymes we have that break down starch can't do anything to cellulose. In fact, very few things can break down cellulose, even termites. They can't do it themselves. They have microbes that live in their gut that break these bonds for them. So cellulose is a very, very tough material. It makes up the wood that we find in trees. Even though there's lots of it out there, we can't consume it. Again, the large carbohydrates that are important to us in terms of nutrition are glycogen and starch. We can make glycogen ourselves, and we can break down starch that we get from plants. We have the enzymes to break both of these. Another interesting thing about cellulose is just how many hydrogen bonds it contains. So cellulose is formed by these long chains of glucose molecules, and again, the glucose molecule alters in its orientation. And that allows each glucose molecule to form hydrogen bonds with adjacent molecules in a different strand. So cellulose is a really tough, resistant substance. Really, really strong structurally. We can build houses out of it, but we can't eat it. But it's pretty amazing to think that it is glucose. I mean, if you go lick a tree, it doesn't taste sweet, but it's all glucose, forming this really, really tough wood. Some animals can take cellulose and break it down and get nutrition from it, but in all cases, at least all the ones I can think of, it's not actually the animal that's doing it, it is a microbe in its gut. So cows will eat lots of grass, contains lots of cellulose in the cell wall. They have a really complex stomach with several chambers, and they bring up the food as cud and they chew that and swallow it again. But despite all that, it's the microbes in their gut that are breaking down the cellulose for them. Another large carbohydrate that's difficult to break down is something known as chitin. Incidentally, if you see CH at the beginning of a word, and the word is derived from Greek, it's pronounced as a hard K, just something you might run into. Chitin is something that's produced by a lot of invertebrates that have an exoskeleton. So they have their skeleton on the outside, basically muscles attached to that skeleton. What you're seeing in B there is a really interesting insect, and I know I'm going off on a tangent again, but this is something called a cicada. It spends its entire life, sometimes decades, underground in the dark, sucking sap from tree roots, and then it climbs up. For one glorious month, it spreads its wings, it flies around and sings. It's the insect that makes that really loud buzzing noise in the trees that you might have heard. In any case, you can see its old exoskeleton there that it's shedding, and that exoskeleton is made of chitin. And chitin has some medical uses. It's actually very, very strong. It doesn't break down immediately, and it can be used to make surgical thread, for instance. So chitin is very similar to cellulose. We won't get into the details all that much, but just note that it's got some nitrogen in it as well. Cellulose doesn't have that, and there's not quite as much hydrogen bonding. Seeing as we're looking at these rather tough structural substances, I'll throw one more at you that's not technically a carbohydrate, but it contains carbohydrate. Peptidoglycan. Peptidoglycan is found in the cell walls of bacteria. In the diagram here, the hexagons that you're seeing, and don't worry about their names, are sugars, they're carbohydrates, and then the little balls are amino acids. So we have chains of carbohydrate that are being held together by short little proteins. That mixture of protein and carbohydrate is not found in us. It's not something we manufacture. If you can damage the cell wall of a bacterium, you generally will kill the bacterium. So there's a lot of antibiotics that attack peptidoglycan. Penicillin, for instance, will attack the enzyme that links those short little proteins together. And lysozyme, which I talked about earlier, I think I might have talked about that already. It's something that's produced by your skin. It's produced within the tears of your eyes. Lysozyme is something that attacks peptidoglycan directly. But again, we don't make peptidoglycan, but it's a common target for antibiotics and a lot of antimicrobials that our bodies produce. Okay, so now our cell has compartments and it's protected from the outer environment by the phospholipids. It also has something to eat. It's got food or energy for long-term storage in the form of triglycerides. If we're talking about a plant cell, we have a tough wall around the outside that's made of cellulose. We're not plants, but that cellulose is important to us because a lot of our food is cellulose. A lot of the plant material we take in consists of cellulose. And of course, that ends up as roughage. Most of that is just going to pass right through your body, but it still stimulates the walls of the intestine to produce mucus and so on. So it's not without value. We can also store additional energy as carbohydrate. So again, plants store it as starch mainly, but we store energy in glycogen. On to our next macromolecule, and this would be the proteins. Pro means first, and these are the most important molecules, I would say, if you're picking favorites. Proteins do all the work in the cell. They make up enzymes, they make up receptors, they do all sorts of things that we'll talk about in a moment. When we think about protein in our diet, we generally think about meat and legumes and so on. But keep in mind that every single cell has proteins in it. Metabolism cannot occur without protein. Movement cannot occur without protein. So what do proteins do? Well, a better question might be, what don't they do? Proteins make up enzymes, for instance, and enzymes catalyze all of the reactions that occur in your body. Those reactions wouldn't occur at all, or they would occur far too slowly to make life possible if we didn't have enzymes. So in this example here, you're seeing a disaccharide, and it's attached to this enzyme, and the enzyme is very specific. It'll only attach to that molecule because of its shape. And what it's doing is it's stressing the bond between those two disaccharides and making it more likely that the bond will break. Proteins can serve a defensive role. So antibodies that bind to foreign substances and foreign cells are made up of protein. And then they'll identify that thing as being something that should be destroyed by your immune system. Storage. So as we've mentioned, we can store energy long-term as fat. We can store it as glycogen in the liver and in muscle cells, but we can also break down proteins as well. And if you look in an egg, you've got the yolk in the middle, which is mostly lipid, and then surrounding that, the white is protein. And both of those are storage structures that store energy for the developing embryo. But we also shuttle energy around as protein in our blood. Transport proteins. So if a cell needs to move something in or move something out of a compartment or exchange stuff with its surroundings, that almost always requires a protein of some sort. It might be a protein that forms a simple channel that allows things to move down their concentration gradient, or it might be an active pump that pumps things in or out of the cell. Proteins can be signaling molecules. So in this example, we've got insulin that's being secreted by the pancreas, and the insulin will find its way into the blood, and it'll be carried by the blood to every single cell in the body. And any cell that has a receptor for insulin will respond to that signal. So it's a way of sending a message everywhere, but only the cells that have the receptor will respond. And what insulin does, of course, is it tells every cell in your body that there's more glucose than we need. You should take it in and do something with it. Store it as fat, just do something with it. Receptor proteins, so receptors for hormones like insulin, but also neuroreceptors. So here we have neurotransmitter that's being released across a synapse between a nerve cell and a cell that will respond in some way. So the receptor is going to send a response into the cell when it binds to that specific signaling molecule. Contractile and motor proteins. So there are proteins that can change shape, and they make movement possible. Movement of flagella and cilia, but also movement of your body through muscular contraction. And that's why meat, which of course is muscle, contains a lot of protein, because there are a lot of these contractile elements within the cell. And it's fascinating how this all works. We'll talk about that in Bio 112. Structural proteins. So we mentioned that in plants, cellulose provides most of the support. So the cell wall is what provides the support. We don't have a cell wall, but we do have something called an extracellular matrix. Outside of the cell, we have a layer of proteins, and they're kind of like cables or bungee cords. They're somewhat elastic, somewhat stretchy, but they support the cell. Most prevalent would be collagen. So we have lots of collagen, makes up connective tissue, but it also makes up that outer layer, that outer extracellular matrix around every cell in your body. So the take-home message here is that proteins do everything. They're even responsible for manufacturing the other macromolecules. So enzymes, remember enzymes are proteins, are responsible for making carbohydrates, and they're responsible for making DNA and RNA, and they're responsible for making lipids. Proteins are made up of amino acids. So amino acids are the building block, the monomer that makes up the protein. Each amino acid has distinct properties and characteristics, and that's determined by the functional groups. And this is the reason I wanted to look at those functional groups. There's 20 different amino acids. There's some other weird exotic ones, but there's 20 important ones. There's 20 amino acids that our DNA codes for instance, which we'll talk about later. There are some that are essential. All amino acids, all 20 are essential, but when someone says this amino acid is essential, what that means is you can only get it from your diet. So some amino acids you can make yourself, some you can't, you have to get them from your food. What happens when a protein is built is, as always, it's a process of dehydration synthesis, and we take individual amino acids and stick them together and form a bond known as a peptide. What you're seeing down the bottom there in green, that is the core of the amino acid, and that part is the same in every single amino acid. And then the part in white hanging off of that chain, that would be the functional group. Different amino acids have different functional groups. That gives each of the 20 amino acids its own particular properties. Amino acids all have the same basic structure. We've got a central carbon, and then attached to one side of that, we've got a carboxyl group, and attached to the other side, we have an amino group. When these are brought together to form a polypeptide, which is a long chain of these things, we always have the same orientation. So we can never have an amino group coming together with another amino group. It's always going to be an amino group coming together through dehydration synthesis with a carboxyl group. And this gives the protein an orientation, as we'll see. Now above that central carbon, we have a hydrogen, and then below it you can see that we've got a variable side chain, a functional group. And there are 20 different things that could be where that R is. So that R is just representing the 20 possible things that we could see there. Note on this figure, it's showing the amino acid in its non-ionized and ionized form. Which form it takes just depends on the pH of the solution. We're not going to worry about that, but usually you'll see it represented in its ionized form. The functional group, shown here highlighted in white, is going to determine the properties of that particular amino acid. The simplest one is glycine. There's just a hydrogen there. The other ones are more complicated. Some of them are quite complicated. Tryptophan has this rather complex side chain with these cyclic structures in it. In fact, the side chain is more complicated than the actual backbone of the amino acid. But all of the ones that you're seeing here are all nonpolar. You can see that there aren't any charges on the side chains. And also we've got a lot of carbon to carbon bonds and carbon to hydrogen bonds. We don't have any nonpolar covalent bonds. So these are side chains that don't interact with water. Incidentally, I'll mention you don't need to know the names of any of these amino acids. Methionine, maybe, but we'll get to that later. I don't expect you to know the structures or the names of any of these. And also note that they can all be abbreviated, so you'll come across those abbreviations. We can also have amino acids that are hydrophilic, meaning they interact with water. If you look at the ones on the top of this diagram, none of them possesses a full charge. so none of the side groups has a plus sign or a negative sign, but we have the uneven sharing of electrons in polar covalent bonds. So you can see a few instances where we have oxygen and hydrogen bound together, or oxygen and carbon. Oxygen is more electronegative than carbon or hydrogen, so it steals the electrons away towards its own nucleus. There's others where we have nitrogen involved, and nitrogen is more electronegative than carbon and hydrogen. I should point out hydroxyl. The hydroxyl group is just written as an OH. There is actually a bond between the O and the H, but it's not shown. The one there that might be confusing you a bit is cysteine, which has that SH. There is a difference in electronegativity between the sulfur and the hydrogen, and that does set up an unequal sharing of electrons. Down the bottom of this diagram, we have the strongly polar side groups, which have a full charge. So the acidic ones have a negative charge, the basic ones have a positive charge. Basically, with the acidic groups, that amino acid, when it's placed into water, it gives up a proton and is left with a negative charge. The basic groups, they give up an electron, or they give up a hydroxyl, I should say, generally, and they're left with a positive charge. We can refer to a protein as a polypeptide. And the reason for that is when dehydration synthesis occurs, we have amino acids being stuck together, and that forms a peptide bond. Poly means many. So a polypeptide has many peptide bonds in it. Note that as we're building a polypeptide or protein, we end up with one end that has a free amino sticking out, an amino group, and at the other end we have a carboxyl group sticking out. So we call the amino end the N terminus, and we call the carboxyl end the C terminus. So any protein is going to have this orientation to it. And actually, if you know the sequence of the protein and you know where the amino end is and the carboxyl end, then you know what order things were stuck together in. So we're always adding new peptides onto the carboxyl terminus. Proteins can be remarkably complex, the most complex of the macromolecules. A polypeptide is the name for a chain of amino acids. And sometimes that's all there is in a protein, just one chain. But a lot of proteins consist of multiple polypeptides. So for instance, hemoglobin, it consists of 4 separate polypeptides, and it's not functional until those 4 pieces come together. Some proteins can consist of hundreds or even thousands of separate polypeptides. And when we talk about the structure, in order to simplify it a bit, we talk about 4 levels of structure, starting with the simplest, which is primary structure, and that is just the sequence of amino acids. Then we have secondary structure, then we have tertiary, which means third, and then quaternary, which means 4th level. And the 4th level is how different polypeptides come together. So we'll talk about each one of these levels of complexity. The primary structure just refers to the sequence of amino acids. So you could have that sequence written out on a piece of paper in front of you. That would be the primary structure of the polypeptide. So here you're seeing a sequence of amino acids, glycine, proline, etc. Note that we have an amino end and we have a carboxyl end. So every single polypeptide is going to have an orientation to it. If we change one of these amino acids, that might have a big effect on the final configuration or structure, shape of the protein. Let's say we took one of the amino acids that was nonpolar and we swapped it with the one that was polar. That would become a very different molecule, perhaps. It might fold in a different way. Now imagine that we had just a straight polypeptide and we threw it into some water. It's going to not stay just a straight polypeptide. It's going to start to fold itself, and all sorts of interesting interactions are going to occur. The first level of interactions define the secondary structure. And secondary structure is the result of hydrogen bonding, quite a lot of hydrogen bonding. So we could put a polypeptide into water and it could spiral itself into something known as an alpha helix. So a helix is a fancy term for a spiral. An alpha helix is a spiral that's formed by hydrogen bonding between adjacent amino acids. The other thing that could happen, imagine that the polypeptide bends back and forth on itself. We could get hydrogen bonding between those folds, resulting in a pleated sheet, kind of like an accordion. And this is known as a beta pleated sheet. So these are common structures that we see in all polypeptides. Note that the hydrogen bonds individually are quite weak, but there's lots of them. So we've got our polypeptide, we've looked at the primary structure and then the secondary structure, and now we're going to add another layer of complexity on top of that. So after the alpha helices and beta pleated sheets have formed, then that structure can be twisted and folded into much larger folds and loops. So this is the folding of the protein, and it's the result of a lot of different interactions. In fact, all of the interactions we've talked about. So we could have a covalent bond. That disulfide bridge that you're seeing in yellow there, that's a covalent bond between two sulfurs on two amino acids that have come close to each other. They might be far apart in the sequence, but the protein has folded itself in such a way that they've come close together. We could have hydrogen bonding, as shown in blue there. So hydrogen bonding is responsible for secondary structures we talked about, but it's also responsible for some elements of tertiary structure. We could have an ionic bond. So what if we have an acidic and basic amino acid and they come close to each other? We're going to have a plus charge and a negative charge, and that interaction will create an ionic bond. And then we can also have hydrophobic interactions. So the non-polar amino acids are going to try to basically get away from the water. They're going to be excluded from the water. All of the polar amino acids are going to interact with water. They might be on the outside of a loop, but the hydrophobic amino acids will cluster together on the inside of a loop to get away from the water. And once they do that, we can also have van der Waals forces acting between those non-polar amino acids. But keep in mind, what determines all of this is the functional groups. So the functional groups are going to determine what happens. And for the most part, it's a passive process. If you throw the protein into water, it's going to rearrange itself until it's in its most stable configuration. That's not always the case, as we'll talk about a bit later. Sometimes the protein needs a bit of help, but the protein is going to take a stable configuration. The final level of structure is quaternary structure. So there are some proteins that only consist of one polypeptide. They don't have any quaternary structure, but the vast majority of proteins do. They consist of more than one part. So you're seeing collagen here, and it consists of 3 polypeptides that are twisted around each other. And that forms a really kind of stretchy, but also quite strong cable. Then we have hemoglobin. I mentioned this before. It consists of 4 separate polypeptides, and each one of those contains a little iron atom, and that's where oxygen binds so that you can transport oxygen through your blood. So just to summarize, we've got the primary structure, which is just the actual sequence of amino acids. Then we've got alpha helices and beta pleated sheets that result from hydrogen bonding. That's the secondary structure. And in tertiary structure, we take all that and we're taking it and bending it into loops and folds. And then quaternary structure, this refers to the fact that most functional proteins consist of more than one polypeptide. So a good short answer question on a test, I might ask you to describe these 4 levels of structure and tell me the chemical bonds or the interactions that are responsible for these levels of structure. So primary structure is the result of covalent bonding. Those are peptide bonds specifically. Secondary structure is entirely the result of hydrogen bonding. And for tertiary structure and quaternary structure, every type of bond and interaction you can think of is potentially involved. So proteins have this really complex 3-dimensional structure to them. Well, what if they lose that structure? That's a process known as denaturation. So imagine here we've got a normal protein, and you can see it's quite complicated. It's got these alpha helices and beta pleated sheets. Those are the straight bits, and it's also folded into some loops. Now that can denature, which essentially means it can unravel. Now under normal conditions in the cell, that shouldn't happen, although in some cases the protein needs a little bit of help, to maintain its shape. But imagine if we had a protein and we heated it, or we changed the pH, or we changed the salt concentration. All of those things could cause the protein to change shape and even unravel. And if it unravels, we say it's denatured. The protein isn't going to work in its denatured state. So if it's an enzyme, for instance, it's got a very specific shape. There's a little notch called the active site where the substrate, the compound it's going to act on, has to bind. And if that shape changes, that enzyme can't work. In the example at the top there, you've got an egg. And remember again, the yolk consists of lipid, but the white is albumin, which is a type of protein. If you throw an egg into a frying pan, of course, the egg white changes. It becomes opaque and rubbery instead of liquid. And what's happened is that albumin has denatured. It's unraveled, and that has very seriously changed the characteristics of that protein. And in the case of the egg white, you can't renature it. There's no way to make it go back to what you started with. Some proteins sometimes can be renatured. Generally in your body, you don't want your proteins to denature. And that's one of the reasons that your body goes to such lengths to maintain an internal environment. There are some parts of your body where we do want denaturation to occur, usually not of our own proteins though. In the stomach, the proteins that you eat are denatured by the very, very acidic conditions. And that means they unravel, and now those peptide bonds that were all hidden in the middle of the molecule are now out there to be picked apart by enzymes that break down the polypeptide into individual amino acids. The primary structure, the sequence of amino acids of a protein, will ultimately determine what kind of shape it can take, because the sequence tells us where we're going to find acidic side groups, where we're going to find basic side groups, where we're going to find hydrophobic side groups, etc. And if you take a polypeptide and throw it in water, it will rearrange itself into its most stable state. So the hydrophilic side chains will interact with water. The hydrophobic ones will get pushed away from the water. They will be excluded from the water, and they will all kind of bunch together in the middle of the protein. But sometimes proteins need a little bit of help to fold properly, and this is accomplished by other proteins called molecular chaperones. And what these molecular chaperones do is they come together and they form almost like a barrel-shaped structure. So the blue structure that you're seeing at the bottom consists of many separate proteins. That's what those kind of subunits are. And the interior of this barrel is all hydrophobic. This chaperone is assembled in such a way that all the hydrophobic side chains are in the center. So we've got a totally hydrophobic little environment inside that canister. So when the polypeptide is put in there, it's not going to immediately twist itself around so that all of the hydrophobic side chains go to the middle. In fact, they're going to go to the outside. And this allows time for kind of new shapes and so on to be formed. So maybe some covalent bonds can form now that will help maintain that structure that wouldn't have been able to occur in an aqueous or water-based environment. So proteins are complicated, right? Well, it turns out they're even more complicated because they can quite often attach to other categories of macromolecules and make something brand new. So we can have lipoproteins, for instance, which are a mixture of protein and lipid. We can have glycoproteins, which are a mixture of protein and carbohydrate. They're mostly protein with a little bit of carbohydrate. We can also call these glycosylated proteins. To glycosylate something is to stick carbohydrate to it, basically stick sugar to it. We can also have proteoglycans, which are also a mixture of protein and carbohydrate, but in this case they're mostly carbohydrate. So they're carbohydrate mainly with a little bit of protein. Glycoproteins serve a number of critical roles in our bodies and in our cells. For instance, they identify cells as belonging to you, and they identify the type of cell. They're also involved in cell-to-cell communication. What you're seeing here is a section of the plasma membrane or cell membrane, and we've got glycoproteins embedded within it. The inside of the cell is at the bottom, the outside of the cell is at the top. notice that we've got sugar monomers attached to the outside of that glycoprotein, and there aren't any inside. Those carbohydrates that are exposed at the surface are recognized by your immune system, and that's one of the ways that the immune system knows that it shouldn't attack this cell because it's part of you. It's also the reason that you might reject a tissue graft or an organ transplant, because the tissue that you're receiving has a different sequence of carbohydrates expressed on the surface. It also helps identify the nature of a cell. So for instance, during embryonic development, a cell will take cues from the cell around it when it's trying to figure out what it's going to develop into. So if we have a cell that is not differentiated or specialized yet, and it's surrounded by heart muscle cells, it will develop into a heart muscle cell, and it's getting that information from these glycoproteins in some cases. Probably the most important group of proteins are the enzymes. So as I mentioned, most of the reactions that take place within your body and within your cells wouldn't occur at all, or if they did, they would occur very, very slowly. And enzymes can speed up these reactions trillions of times. Enzymes act as catalysts. A catalyst is something that accelerates a reaction, and they can be reused over and over again. They're not destroyed during the reaction. Enzymes are quite picky when it comes to what they do. So the example at the bottom here, you've got an enzyme that's binding to a substrate. The substrate is the compound or substance that it's going to act on. And notice that there's kind of a notch in the enzyme, known as the active site, that has a very specific shape, and it will only bind a certain molecule, or just a small handful of molecules. It will bind to that substrate, and in this case, it's going to split that substrate and break off monomers. Enzymes are very large molecules, as you can see in this diagram. Every green little sphere represents an individual atom. This enzyme has a cleft, known as the active site, and that's where the substrate is going to bind, and it has to have the right shape. It starts out as a fairly loose fit, but then the enzyme will wrap around it. Once it wraps around it, there has to be a very tight fit. There has to be the appropriate alignment of charges and so on. And if that doesn't occur, then the enzyme will spit this back out because it's not the right substrate. So that's called an induced fit when the enzyme wraps around the substrate. This particular enzyme is something known as hexokinase. You don't have to worry about that, but I'll just mention that a kinase is something that will phosphorylate something. What I mean by that is it'll take a phosphate and stick it onto the substrate. So this enzyme, its job is to take a phosphate group and stick it onto glucose, which is one of the steps that occurs during the breakdown of glucose to release energy. So let's take a look at a fairly typical reaction that might be catalyzed by an enzyme. What we've got here is we've got one molecule, BC, that's going to interact with molecule A, and the B portion of that BC molecule is going to be transferred to A. Now this might happen naturally without any help, but only if those two molecules collide with each other in exactly the right way, with exactly the right energy. What the enzyme will do is it will grab onto these two molecules, and it's got a very specific active site that can hold those two molecules. So the first step in this reaction is initiation. The reactants are going to bind to the active site in a specific orientation, and what's going to happen next is the enzyme is basically going to line those bits up in exactly the orientation that needs to occur in order for the reaction to occur. Then we've got what's called the transition state. Now what's happening here is the bond between B and C is being physically stressed. So the enzyme will change shape slightly. It'll put strain on that bond, and also it's bringing A close to B so that we can get a bond forming between A and B. Now what this does is it just greatly enhances the chance of the reaction occurring. It's making conditions absolutely optimal, and that lowers the activation energy, the amount of energy that's required to be put in for the reaction to occur. And now we've transferred B over to A to create AB, and that's going to be released. It's going to be released from the active site. The active site hasn't changed, the enzyme hasn't been damaged, although they do eventually wear out, but the enzyme can be reused over and over and over again. And this reaction that you just saw is one that's fairly common, where one subunit is transferred to something else. So for instance, there are kinases that I just talked about that transfer phosphates from one molecule to another. So in the process of cellular respiration, where we liberate energy from our food, what happens is phosphate groups that have been stuck to food substrates are pulled off and stuck to ADP to make ATP. There's a few reactions during the whole process where that occurs. So how might we control enzyme activity within your cell, and how does your cell control it? Well, there's two broad ways that this might occur. So let's go back to the enzyme we were talking about before. Remember that that enzyme uses glucose as its substrate, so it binds to glucose and then adds a phosphate to it. Let's imagine that we have another molecule that has a similar shape. Now that molecule might also fit into the active site. It might not fit very well, and the enzyme might spit it out right away. And even if it does bind, the enzyme probably can't do anything to it. But that other molecule is kind of an imposter. And even for the brief little period of time that it's bound to the active site, it prevents the real substrate from binding. So let's imagine a case where we have a cell and it has a tiny amount of the substrate. And then we add an overwhelming amount of the competitive inhibitor, the imposter molecule. The enzyme is going to spend all its time dealing with that competitive inhibitor, and it's pretty much going to shut down the reaction. Again, just keep in mind the competitive inhibitor isn't there for very long. Usually it's going to be released right away, but because there's so much more of it, it's going to block the reaction. There are a number of pharmaceuticals that work this way. An interesting example, I think, is what happens if you have alcohol poisoning, specifically methanol poisoning. There's an enzyme in your cells called alcohol dehydrogenase, and it's the enzyme responsible for the first in a series of steps to break down ethanol. Usually ethanol is what we drink. It's in beer and vodka, everything else. It will also bind to methanol. Methanol is quite harmful. You should never drink methanol. Methanol is broken down into some compounds that are rather toxic, and they're especially harmful to the rods and cones of your eyes. They can cause blindness. If someone comes into the emergency room and they have consumed methanol, what they immediately do is they get the person fall down drunk. Basically, they will hook up an IV with ethanol, with vodka, essentially. They'll give them so much ethanol that the ethanol will compete for the active site on that alcohol dehydrogenase. Methanol, fortunately, is a very small molecule, and if it's floating around in your system for a while, it will be removed by your kidneys. But like I said, if it's broken down by alcohol dehydrogenase, then it becomes something rather toxic. So the goal is to overwhelm alcohol dehydrogenase until you can get the methanol out of your system. Now, the other way that we can regulate enzyme activity, and this is the method that's more commonly used by your body, is known as allosteric regulation. Allosteric literally means elsewhere, somewhere else. So in this case, we're not regulating the activity of the active site by blocking the active site. Instead, something else is happening somewhere else on the enzyme. So most enzymes will have a little cleft elsewhere that doesn't bind to the substrate. Instead, it binds to a regulatory molecule. And when the molecule binds to that spot, it changes the shape of the enzyme. Typically what happens is it causes the active site to close, and then the enzyme is shut off. There are several other factors that can control the rate of enzymatic activity. Just like any chemical reaction, if you increase the temperature, that means you increase the speed at which molecules are moving, and they bump into each other more often. So it means that reactions are going to occur more often. And if you look on the first graph there for temperature, you can see that if we start at a low temperature, 10 degrees, that would never happen in your body, of course, but let's say we're doing this with enzymes that we've extracted and we put them in a beaker. At 10 degrees, we have very little activity, but then as we increase the temperature, it gets higher and higher and higher until we get up to about 37 degrees. 37 degrees is body temperature for humans and other mammals, and our enzymes have evolved to work best at that temperature. Now, when you go above 37 degrees, somewhat surprisingly, the activity drops off very dramatically. And if you think about it, it's not that surprising because enzymes are proteins. And when we get above 40 degrees, the enzymes denature. So it's really important that you don't get too warm. If your internal body temperature gets too warm, your proteins, your enzymes, for instance, will denature and they'll stop working. There are some chaperone proteins, they're called, that can bind to enzymes and help to keep them together at higher temperatures, but even that has limitations. pH affects enzymatic activity quite a lot. We have many enzymes in our body, and they prefer different pHs. So most enzymes prefer pH around neutral, around 7. That's what we're most likely to encounter in the cytoplasm of the cell. But we have compartments within the cell, lysosomes, for instance, that are very acidic, and they contain enzymes that only function under those conditions. That's actually a way to protect the cell, because if those were to rupture and release the contents into the cell, the rest of the cell is neutral. The enzymes that are found within lysosomes are really good at breaking down pretty much everything, and they won't digest the cell. We'll talk about that later. It's kind of an interesting process. In the digestive system, the stomach is very, very acidic, and pepsin, which works in the stomach, prefers very acidic conditions. And there's some other parts of the body that are slightly alkaline as well. The substrate concentration is important as well. So in that last graph, C, what you're seeing is additional substrate being added. So we have more and more substrate being added along the x-axis. And notice that the enzymatic activity increases, or the reaction rate increases, as you would expect. There's more molecules to be involved in reactions, but we get to a saturation point. And that's the point where the enzyme is basically overwhelmed. So the enzyme is receiving inputs into the active site as fast as it can deal with them. If you want the reaction to occur faster than that saturation point, you just have to add more enzyme to your reaction. So here's a nice example of the effects of pH on the functioning of different enzymes. We've got 3 enzymes here that are very important in your digestive system. Pepsin and trypsin both break down protein, but they're found in different areas, or at least they're active in different areas. So pepsin is active in the stomach. In order for it to have its correct folding, it needs to be in a very, very acidic environment. And the stomach is incredibly acidic, less than 2. So more acidic than battery acid. Remember that the stomach is acidic so that the proteins you eat will unravel. And pepsin will take those very large molecules and break them into smaller chunks. Once the contents of the stomach empty into the intestine, the pH is going to change. The pH is going to become actually slightly alkaline in some places. Trypsin then takes over. Pepsin becomes deactivated, but trypsin has its correct folding at neutral or slightly alkaline conditions. And then it will take those chunks and break them down even further. Amylase, you can see, prefers a near neutral pH. Amylase breaks down starch. So different things can happen in different parts of the digestive system, and what enzymes are going to be active is determined by the pH. Likewise, in the cell, as I mentioned, we've got compartments within the cell, organelles. Those organelles have different pHs so that different enzymes will be active in those different areas. And if an enzyme escapes from a compartment and ends up somewhere it shouldn't, it won't do any damage. So back to our cell. If you remember, we were building a cell. That was the premise of this lecture here. And we've looked at 3 different groups of macromolecules, and these provide the cell with structure, something to eat, and also the machinery to do all the work that needs to be done. So I've represented proteins here as little lightning bolts because they do all the work. We have one last thing we have to do, and that is we have to provide the cell with blueprints that it can pass on to the next generation of cells or the next generation of individuals. To do that, of course, requires nucleic acids. Nucleic acids store information, and the information they store is information needed to make proteins. So they're responsible for storing that information. Proteins are made, and then the proteins do everything else. The proteins make carbohydrates, the proteins make lipids, etc. Nucleic acids are responsible for storing and transmitting genetic information from generation to generation. So the nucleus within a cell contains DNA. And people quite often compare the nucleus to the brain of the cell. I don't really like that analogy. I think of the nucleus as the library of the cell. The nucleus is a massive library filled with lots of books. And each one of those books is a gene. And each gene is the manual for manufacturing one specific protein. So DNA is the storage device for that genetic material. These are the blueprints. Ribonucleic acid, RNA, is going to carry information from the nucleus out to the cytoplasm, out to a ribosome, where it can be decoded to make protein. So let's have a quick look at the structure of nucleic acids. Like other macromolecules, they're made up of monomers that are stuck together one at a time through dehydration synthesis. The monomers are known as nucleotides, and you can see one here. So a nucleotide consists of three parts: a phosphate group, a sugar, a pentose sugar, which just means it's got that pentagram shape to it, and also a nitrogenous base, which, as the name suggests, contains nitrogen. We have a number of different nitrogenous bases. So in DNA, we have cytosine, thymine, adenine, and guanine. In RNA, instead of thymine, we have uracil, and then we have the other three. There are two types of sugar. In DNA, we have something called deoxyribose, and in RNA, we have ribose. You might have heard of Watson and Crick. A lot of people mistakenly think that they discovered DNA. They didn't actually. DNA was first isolated back in the 1800s, but they were able to figure out the structure of DNA. So they looked at a lot of research by other people, pieced it all together, and gave us the model that we have today of DNA. And they referred to DNA as a double helix. A helix is a spiral, so basically we have two strands wrapped around each other. And if you're looking for a fairly short, interesting science book to read, they actually published a book called The Double Helix. And here's the structure of DNA. So again, it consists of two strands that are twisted around each other. On the far right, you can see the space-filling model. That's about as close as we can get to, a realistic interpretation of what it looks like. But on the far left is how you're more likely to see it in a textbook. The blue represents the backbone of the molecule, which consists of the phosphates and the sugar. And then the nitrogenous bases are in the middle. And it's kind of like rungs of a ladder. So the nitrogenous bases are held together by hydrogen bonds. That's what the dotted red lines are. In the middle here, you can see just a close-up on a few of those rungs. So we're seeing 4 rungs in the ladder. Again, the backbone would be the phosphates and the sugar. And then in the middle, we have our nitrogenous bases, which are cyclical structures. Now, one thing to note is that the bases always line up in a very specific way. So adenine and thymine bond together, and guanine and cytosine bond together. And note that G and C actually make 3 hydrogen bonds, A and T makes only 2. So we always have this kind of arrangement holding the DNA molecule together. Remember, hydrogen bonds are quite weak, but there's lots of them, so this is a stable molecule. But at the same time, an enzyme called RNA polymerase can come in and just unzip a few of those rungs, just a few at a time, and read the information. And then as that enzyme travels down the middle of the DNA molecule, it will zip back up behind the enzyme. So how does DNA differ from RNA? In a few ways. So first of all, function. Remember that DNA stays put in the nucleus. It's a permanent repository for information. And I should point out that you do have a bit of DNA outside the nucleus. You have DNA within your mitochondria. In each cell, you have about 20,000 genes. 37 of those are in the mitochondria. So not a whole lot, but those genes are quite important. The RNA molecules serve a few different functions. Some of them are found in ribosomes, where they make up part of the structure of the ribosome. But the most important ones make up a molecule known as messenger RNA. Messenger RNA is a copy of the information from one gene in the nucleus. So that information will leave the nucleus. It goes out through pores in the nuclear envelope. That's the double membrane that surrounds the nucleus. Finds a ribosome, and then that is translated into a protein sequence, a sequence of amino acids. So DNA stays put. RNA transfers the message to a ribosome. Now there are some structural differences as well. So first of all, where do the names come from? Ribose is a sugar that makes up RNA. Deoxyribose, or technically 2-deoxyribose, is the sugar that's found in DNA. And there's only one slight difference there. Notice that in DNA, we're missing an oxygen on the second carbon. Another difference is that RNA is single-stranded, whereas DNA is double-stranded. So again, DNA is a double helix, RNA is a single helix. RNA is a more fragile molecule because of that. It can be broken down quite readily. Now there are a few instances where we might see short little bits of double-stranded RNA in our cells, but we won't get into that. Also, it's kind of interesting in that double-stranded RNA is quite common in many viruses. Again, though, for human cells, RNA is almost always going to be single-stranded. Finally, we have a difference when it comes to the bases, and it's a small difference. In RNA, we have something called uracil, and in DNA, that's replaced by thymine. They are quite similar. Let's talk very briefly about how we name the bits and pieces that make up nucleic acids. For instance, when should you use the term nucleoside and when should you use the term nucleotide? A nucleoside is a sugar and a base, so we're not including the phosphate here. Normally you would use the term nucleoside if you're talking about a complete molecule of RNA or DNA and you're listing off the sequence of bits. You're listing off the sequence of nucleosides. And that's just because you don't care about the phosphate. The phosphate doesn't change the property of that sequence. But if you're talking about the individual pieces, the monomers, and you haven't stuck them together yet, then we use the term nucleotide. So nucleotide refers to the sugar, the base, and the phosphate. Now both of the examples I've given you here are RNA nucleosides and nucleotides. This one here would be a DNA nucleotide. We know it's DNA because there's an oxygen missing where the arrow is, and we call it a nucleotide because we're including the phosphate in our discussion. When we're dealing with nucleotides, we might also want to indicate how many phosphates are attached. So the nucleotides start off with 3 phosphates attached. When we combine them together to form a molecule of RNA or DNA, 2 of the phosphates are cut off. Now this is a molecule here which you should recognize at this point, ATP, adenosine triphosphate. The adenosine comes from the name of the base at the top. That's the 2-ring structure at the top with lots of nitrogen. The T stands for tri, 3, and that's because there's 3 phosphates attached. Incidentally, this is an RNA nucleotide, and we know that because there's an oxygen here. So ATP is used for many things. We've talked about how it's used to shuttle energy around. It's also used to build RNA, and it's used as a signaling molecule. It does lots of stuff. Now, if we take one of the phosphates off, then we have adenosine diphosphate, ADP. If we take 2 of the phosphates off, then we have adenosine monophosphate, AMP. And in fact, there are some chemical reactions that use ADP to shuttle energy around. A phosphate is pulled off to make AMP, and then that's recycled. But we won't get into that stuff. Usually it's ATP being converted to ADP and back again. There's one more thing we should talk about briefly, and I'd like to talk about it in a lot more detail, and I will later, but that is what's known as the central dogma of molecular biology. Now, molecular biology is the study of DNA and how DNA is used to make proteins and how this whole system functions. And that's my background, so I would very much like to talk more about it, but I'm going to keep it really, really short. The central dogma suggests something very simple. DNA is used as a template to make copies of itself. DNA is used as a template to make RNA. RNA is used as a template to make proteins. So DNA can split into two strands, and then enzymes will come along and copy the existing strands to replace the second strand. And that's a replication of DNA. And that happens whenever a cell is getting ready to divide. Transcription is the process whereby the information found in DNA is used to make a copy in RNA. And finally, translation is where the information in RNA is used to make a protein. So the information that's found in DNA and then RNA is a code. It's a code for amino acids. So each amino acid can be coded for by 3 nucleosides. The other thing to note is that this central dogma applies to all living things. Now, viruses, some of them can do things a bit differently. They're also not usually considered to be alive. Also, a gene is a unit of DNA, a segment of DNA that codes for one polypeptide. So you have over 20,000 genes. They code for different proteins. And it's a bit more complicated than that, but that's as far as we need to go for the moment. Now, because this applies to all living things and because all living things follow the same code, you can take a piece of DNA from a bacterial cell and you can put that into an animal cell, and the animal cell will decode that DNA and use it to make a protein. Another really nice example, going the other way, the production of insulin. So insulin is a protein, it's a hormone, and before we were getting into genetic engineering and so on, the only way to get insulin in large quantities for people that needed it, people that were diabetic, was to collect it from horses. That was a really labor-intensive job, and also horse insulin is slightly different than human insulin. It doesn't work quite as well as it could. But what we do now, and we've done this actually for decades now, is we've taken the gene from human cells that codes for insulin, taken that gene, put it into bacteria, and now the bacteria will read that information and they will convert that information into a protein. Insulin is not something they need. It's not something that would normally be there, but the information is there and they have to decode it, basically. They have to decode it and use it to make protein. And then we can grow up insulin in bacteria and in yeast cells. We can grow it up in huge amounts and purify it, and we get pure human insulin. So to sum up, once again, we've got DNA, double-stranded molecule, stays put within the nucleus. It can be unzipped just a little bit at a time, and the exposed nucleosides can be read by an enzyme called RNA polymerase that will make a copy of that message in RNA. Now remember, DNA and RNA are both made up of nucleosides. So we are transcribing or copying the information from DNA to RNA, but we're not changing the language. I like to think of the monomers as kind of an alphabet. Now the RNA is going to leave the nucleus and go out to the cytoplasm. RNA is made up of nucleosides. Proteins are made up of amino acids. We're going to use the information in the messenger RNA to manufacture a sequence of amino acids. And we're changing the language here. We're changing the alphabet. We're going from nucleosides to amino acids. And how this works is that the nucleotides form what are called codons. And this consists of just 3 nucleosides. 3 nucleosides will code for an amino acid. So all of the 20 different amino acids can be coded for by a sequence of 3 nucleosides. So we're done. We have a cell that has structure, it has something to eat, it can do things, and it can also store and transmit information. The DNA stores information on how to make protein, and then the proteins do everything else, including replicating the DNA and transcribing the DNA into RNA. And here's what an actual complete cell looks like. And finally, here's our terminology list for topics 2 and 3. It's pretty long. There's 2 slides here. But these are words that I'm hoping you were already familiar with. This was a review. This basic chemistry and a tiny bit of organic chemistry, these are all things that you should have learned before you were admitted to this course, but I want to make sure you have a chance to catch up if you didn't, or if you've forgotten it. I want to make sure we're all on the same footing before we go forward. So if it doesn't make sense, make sure you review this. You will be tested on this stuff. If it still doesn't make sense after you've gone over it a few times, definitely contact me, ask me lots of questions, and I'll help you out with it. There's also a really extensive study guide that I've written and put up on Moodle. Go over that as well. And just to note, a few people have asked, the study guides aren't everything you need to know. What you need to know is what's in the PowerPoints and what I've talked about. Specifically, you need to be able to define and explain all of these terms, and you need to be able to answer questions about those terms.